Here I have a plate and I've got a sample where I've ground up seed material and it's now a flour. In the plate there's 2 blanks. Those are for standards in the lab, so I always put those in first. This is the first sample I'm putting into the plate. I'm going to handle it individually away from all other tubes, and every time I do this it'll be the same. Tubes that are in the plate are sealed, so it's okay to handle materials around the plate. Just make sure that your tubes are capped when you're doing that. And so I'm going to write down the weld position, which happens to be A1 as the first position for this material. Each plate should have material from only one crop species. And you'll have marked your plate, so you would record this number to know what plate this sample is going into, and then for this number you would record that plate number and then also the weld position. You might also affix barcode to this, in which case that number would be recorded against this, and if you've got double barcodes you can actually stick the other one onto this packet so it's physically present if you need to know. At this point you'll transfer about 20 to 30 milligrams of ground material into the tube. So you're going to use a spatula or scoop to do that, and that material you'll scoop up and transfer it into the tube. So that's probably about 10 or 15. You can see here that this can be a messy activity. That's why you don't want other tubes that are open near it, or you can get contamination. And so getting a little bit more, I'll move it around in the packet. Yep. And that will then go in, tamp it down. That's about the amount of material that you want in there, just at the bottom of the tube, 20 to 30 milligrams, not very much. You don't want this tube to be very full, it's just at the bottom. This in fact might be a little bit too much material, or just a little bit more than desired, in which case you can decant back a small amount of material there and end up with the amount that you want. That was actually too much, which is part of the finickiness of dealing with ground flour material. And so scooping again to get the right amount. Then that looks about right. So we will then cap this, and that will be in position with everything recorded. You'll then do this for all of the other positions in the plate, which are 94. In between doing this, you need to wash thoroughly your scoop so that you don't contaminate one sample to the other. So do that with isopropyl alcohol and a disposable wipe in between samples, and make sure it dries so that you don't get samples sticking to it. So again, it's just a tiny amount of sample, 20 to 30 milligrams. The consistency needs to be that of flour, and then you need to make sure that that goes into the right weld position. So this is A1, that's all been recorded against that number, including information about what plate it is, because you'll have multiple plates, and you don't want A1 to end up in D1 or A10 or some other place. It needs to be in the well that it's indicated for. Otherwise the data won't align with the sample.